My name is Abhirami Aiswarya Jayaraman. My matric number is 23DBT20F1035. My class is DBT3B. Let's start the stimulation. Before you start the lab stimulation, choose level 3 and then start stimulation. Context: SDS page is a discontinuous electrophoretic system which is commonly used as a method to separate proteins with molecular masses between 5 and 250 kDa. SDS page is commonly used to obtain high-resolution separation of complex mixtures of proteins. The method initially denatures the protein that will undergo electrophoresis. Materials, reagents, chemical that can be used in this experiment is 4 times SDS sample, wild type cell culture, pancreas cell culture, edit 4 cell culture, edit 7 cell culture, protein ladle solution, 1 times running buffer. All of these are the solution used in this experiment. Micropipetting equipment used to measure small volumes. As you can see, various type of pipette and tip box. Here are the empty tubes which is used to hold solutions, and then electrophoresis equipment which is used to sort molecules by size and charge. And we also use other equipment as microcentrifuge, biohazard waste and heat block. Prediction: in this simulation, we'll use SDS page to separate proteins to cellular extract which contain all proteins of cell. The extracts we are interested in come from 2 crisp edited macrophage cell lines. Let's see positive control. Which of the following do you think will be a good positive control for this experiment? Then let's see negative control. Which of the following cell line will provide an effective negative control for this experiment? Now let's identify controls and samples. Those are: read through the protocol, collect cells, add sample buffer, denature the proteins, contribute the sample, load the gel, conduct protein electrophoresis. Now let's start the stimulation. The first part will be collect the cells. Firstly, increase the heat block temperature to 95 degrees Celsius. Set a micropipette to 30 and attach a tip. And then draw up the wild type cell culture. Now close the wild-type cell culture. Later, dispense the wild-type cell culture into the wild-type cells tube. Then, repeat the procedure to transfer 30 of the pancreas cells culture. Now repeat the procedure to transfer 30 of the edit 4 cell culture. Then, repeat the procedure to transfer 30 of the edit 7 cell culture. The second part will be add sample buffer. Firstly, set a micropipette to 10 and attach a tip. And then draw up 10 of 4x SDS sample buffer. Then close the 4x SDS sample buffer. Now dispense the 4x SDS sample buffer into the wild-type cells tube. Repeat the procedure to dispense 10 of 4x SDS sample buffer into the pancreas cells tube. Repeat the procedure to dispense 10 of 4x SDS sample buffer into the edit 4 tube. Then repeat the procedure to dispense 10 of 4x SDS sample into the edit 7 tube. The protein ladder already contains SDS sample buffer. The third part is denature the proteins. Firstly, move all of your 4 sample tubes to the heat block. Set the heat block timer to 10 minutes and press start. Then, remove all of your sample tubes from the heat block. The fourth part will be centrifuge the samples. Firstly, move all of your sample tubes to the microcentrifuge. Then, briefly spin the tubes. Then, remove the tubes. The fifth part will be load the gel. Firstly, place the polyacrylate gel in the electrophoresis box. Then, fill the gel cassette with 1 times running buffer. Now, remove the comb. Fill the outer gel electrophoresis tank with 1 times running buffer. Set a micropipette to 20 and attach a tip. Now, draw up the protein ladder. Move the micropipette to the first line of the protein gel. Now, dispense the protein ladder into the first wall. Dispense 20 of the positive control into the second vial of the gel. Now, dispense 20 of the negative control into the third vial of the gel. Then dispense 20 of the added 4 cells into the fourth well of the gel. Lastly, dispense 20 of the added 7 cells into the fifth well of the gel. The sixth part will be conduct protein electrophoresis. Firstly, cover the electrophoresis box. Then, connect the red and black cables to the power supply and the lid of the gel electrophoresis box. Then, turn on the power supply. Now, set the voltage to 150 volt. Then, set a timer to 60 minutes. Then, start the power supply. Now, wait until the time has elapsed. Now, turn off the power switch. Later, unplugging the electrodes. And now, inspect your gel. The next part will be the results. The next part will be reflection. The reason we use SDS in this essay is why are samples heated before loading them on SDS gel? Why might you rather want to run the SDS gel electrophoresis at low voltage than a high voltage? SDS sample buffer contains glycerol which has a higher density than running buffer. Why glycerol included in SDS sample buffer? In this experiment, you aim to detect the presence or absence of a specific protein produced by the CCR5 gene. What proteins were separated by size during the SDS PAGE gel electrophoresis. The final part will be the summary. In this simulation, you treated your cell culture samples with SDS sample buffer and heated them to ensure cell lysis and the denaturation of all cellular proteins. SDS code to denature protein in negative charge. After loading your samples and controls on a protein gel, you separate proteins according to molecular weight by applying an electrical current. Since the proteins are invisible, look for the blue dye front and protein ladder reaching the bottom of the gel as indicators of when to stop the electrophoresis. Here the end of simulation. That's all from me. Thank you.